Hey guys, Steve here, Ponics. Today we're going to talk about
Growing with fishes. Growing with fishes.
Hey everybody, welcome to the Growing with Fishes podcast. We have another returning guest today. We have Brandon Rust, who's provided a ton of great knowledge and information on the show previously, and we're excited to have him back on today. Thanks a lot for joining us, Brandon.
Thanks for having me, man. It's been a minute since we've talked.
Yeah, it's been 100 episodes, something like that, since we've had you on. It's nice to get you back on. We try to get people that we like and people that really know their shit on the show a couple of times pretty regularly. So I appreciate you coming back on.
Yeah, no problem.
I know you've been real busy doing all kinds of stuff this winter. I know you did a big overhaul on your website. You want to maybe start with that because you got so many great tools on there that are, people can check out for free and help with their grows and stuff like that.
Yeah, absolutely. Yeah, we relaunched the website. So previously we were Bokashi Earthworks. When I went to try to file a trademark, it was denied because I couldn't use the name Earthworks. So we rebranded as Full Crop Sciences with one L, like fulvic acid. And we have, our products on there, the Microbe Plus, Humate Fertilizer, some amendments, and then the microbe spores. We have a nutrient calculator for Logan's Labs as well, which is pretty cool. And then on which you have to pay to access that, but I'm gonna try to make that free in the future, but I have to get with the developer that built it for me. And then also what's really cool is if you sign up on the newsletter on the website, regardless if you buy anything, you'll get access to all of our social platforms, the YouTube channel, the Discord, the Dropbox with science white papers, and it follows up with, free guides for different types of things, fixing purple stems, lighting guides, dry cure guides, all different types of stuff that are relevant to cultivation.
And you got a lot of really kick-ass breakdowns on the science of different things in your blog as well. The
Yes,
things
those are
I've
all the
liked
guides.
regularly. I've
Yeah.
regularly sent people to your website to check out and learn about different things.
Yeah, so what I'll typically do is I'll do like a pre-recorded podcast. Usually the pre-recorded part is usually 5 to maybe 15 minutes long, and then I do a Q&A, but it's always on the same subject matter of what I'm putting into the blog. So like if you're wanting to learn about amino acids, there's a podcast and there's a free guide that you can look through and learn about, how they chelate nutrients and how they act as biostimulants and all the different things that amino acids do is, saving plants energy, and there's so much different stuff on there.
You got all kinds of great stuff on KNF stuff and the different microbes. It's really great.
Yeah, and what's really cool too is so what we did is we trained our AI model on I basically trained it on all of the science and stuff and uploaded things like my Dropbox and all kinds of stuff like that. And then what we also do is when we make these, we link links to all of the science white papers as well. So you can go on there and if you want to explore more of the science, like the peer-reviewed research, it's there's links in those guides as well.
Awesome. Yeah, no, it's a great resource for people that aren't aware of it. And you have such good breakdowns of many of the different topics. It's a really good resource for people to check out for sure.
Yeah.
I know you've been working on getting an operation going there now in Oklahoma as well. Do you want to talk a little bit about your dispo and grow that's coming down the pipeline here soon?
Yeah, well, for any of the people that are unfamiliar with me, I've been cultivating for over 2 decades since 2002 professionally. I was brought on by some OGs, and so yeah, since I've been in Oklahoma, I've ran huge facilities, I've consulted. We had a 40-acre farm at one time, which we actually failed, but it was a learning experience, so I don't take, it's not a failure if you take something away from it. But we went huge, right? And so it was me and my partner from Cali that were doing all the work, 12-hour days in the mixed light greenhouses, and we were relying on our business partners to go pound pavement and get the deals done with large quantities that we were producing, and it's quantities that we had never personally produced before, and we didn't have that type of market, so it was relying on the relationships with dispensaries and distributors and stuff like that, and they just weren't good. They weren't from the space, right? And so it was a lot harder for them to, I think, like mesh with the culture. Um, and so when I had an opportunity, I was like, I'm not going to do this again. It's too much of an invest. Like the risk on investment in the space is just too high from a business perspective. And so I just worked in a consulting capacity for a long time. Didn't have any my hands directly involved in any type of licensed facilities or anything like that. But my friend Alex, who he was the first person that I hired when I came out here. And I was cultivation director for a bunch of investors at another facility when I first got to Oklahoma. I taught him the back end, and then he ended up doing sales. He ended up getting a dispensary with some business partners, and we actually found a spot that we could be vertically integrated. So we could cut out the logistics of having distribution. We could rely on our own shop to do the distribution, and it's not a large spot because I've worked in like large capacities, hundreds and hundreds of lights, and it's like the logistics and the efficiencies and being able to like run that and manage workforces and manage inventory and manage the quantity is a lot. So what I did is I kind of went back to my roots on this, and this is the whole facility's 22 lights. So we have 14 lights and three across three different flowering rooms. So we have a 6 light flowering room and two 4 light flowering rooms. We have uh, a mom room, we have a veg room, we have a like a warehouse area. We have a place where we can dry and also trim and store everything. So it's all temperature controlled. And then we have the front office and the dispo and everything else. And it's all going to be for in-house. We don't have to worry about the logistics of trying to wholesale, and we're not putting ourselves in that market. So we could sell, 30 or $35 eighths out the door all day long, be profitable, keep our overhead low, do living soil cultivation practices, which reduces our overhead in the management of the daily operations. And so I just went back to our roots. We used to run, houses that were, maybe 12, 13 to maybe like 20, 25 lights, and that's we might have multiple places like that, but that's where we were back in the trap days, and we weren't relying on distributors. We were all we were able to break our product up and sell pounds and half pounds and get better deals. And so I was like, that's the business model that I want to have. So we had an opportunity. We found like a 2,800 square foot place that was previously semi built out for cultivation, but it didn't look like they knew what they were doing. And the rent on the place was it was only like 1,450 bucks a month. So it's like, dude, we can get this all set up, we can lace it up, and even if it takes us a while to get all of our stuff from the state, the overhead won't annihilate us. So it's been over a year. The place has been built out for over six months. It's been over a year since we started, and we just got word that February 20th will be the date that we'll have our final interviews done, because we have our COOs, the OMMA stuff. Just everything's finished except for the Department of Narcotics, and they're just taking their time. So we'll be open right after that. So in March we should be opening up, and we're going to start getting plants in there. We've got a hemp license as well there. We'll also, have our tobacco license, so we'll sell other stuff, and then people can come in there and get, whatever their cigarettes or whatever. I mean, I don't
That's
smoke
cool.
cigarettes. I, vape. I shouldn't, and I do it off and on, like. But yeah, man. It's cool. It's gonna be a small system. We're not gonna be like chasing shit all the time. I'm just gonna have standard in-house flavors. We'll have ChemDog on the menu. We'll have some of my stuff on the menu, and those things will just kind of, stay the same. We'll do a few things for the menu, like probably 6 in-house flavors that we just do consistently. We know how to grow them. They're done really well. We're trying to create consistency and not just trying to like, oh, like find the new hype thing or whatever.
Cool.
Just like I used to, man.
That's cool that they allow the tobacco licensing with the cannabis. Finally, you can get your blunt wraps with your weed. That's pretty great. I know that my favorite when I lived in Oklahoma, at the end of my street there in Tecumseh, there was a combination butcher shop and tobacco store, and it was like, how the fuck does the zoning work on that? We get so much shit for the weed licensing,
Well,
which is
the
kind of
zoning
funny.
is the same. That the license is just a license. It's just that you apply for it at whatever location you're going to be selling at, and it's not like they come in like, they might come in there once a year and like, just make sure that you're um, operating properly or whatever. But it's like, yeah, you can have tobacco products and things that you might have at like a regular smoke shop there too. Have some pipes and bongs and trays, papers, all that kind of stuff.
I just thought it was funny because we couldn't get a meat processing license to process our fish for aquaponics facilities because of the cannabis licensing. So, but tobacco's fine, there it's just one of those stupid loopholes they haven't fixed yet.
Yeah, it doesn't matter. It doesn't need to be fixed. They don't need to make any more. They don't need to keep making regulations and stuff like that. That shit's just like stupid. I'm tired of it. Just
Yeah,
less
I still,
regulations. Less government is good government.
no, I agree. But you still can't do meat or dairy, unfortunately, because they're regulated differently, which sucks. Um, the I also like outdoor seasons coming up. I know you have those awesome NutriPots. So you want to mention those too because they're great for people that are going to get started and then transfer those to outdoors.
Yeah, so those are really, it's not logistically efficient to manufacture those manually. So we haven't been making them or selling them. But this year, what we will be doing is working on getting the machinery to do mass production. So, right now what we're currently looking at, like tomorrow I'm going to Hera to go look at a farm, and we're looking at these old like a lot of cannabis farms that are selling. We're trying to find a place that already has the infrastructure built, but I'm not going for a cannabis farm. What I'm trying to do is take advantage of a FSA loan, farm loan, and business operating loan from the USDA, and then take advantage of the regenerative agriculture grants. And I want to start a organic produce farm. So I'm trying to find some place that already has the infrastructure as far as like greenhouses and building set up so I did the math, and essentially I could get a loan and I would be saving over $2,000 a month if I moved my business and my living situation. Like I would rent out my house or do something with this property here, move to the farm, operate a produce farm, operate full crop science and my other businesses out of the same location so I create better logistics for all and have an umbrella. And besides, I'm putting everything in a irrevocable trust anyway. So that way all my assets and my companies and everything are in the trust, nothing's in my name. And the thing is, like I was looking at the actual finance behind it and the at the interest rates for the FSA loan and the business loans, I'd be paying basically $400 less than I do on my current lease for my office. And then if I actually moved there, I'd be saving an additional couple $1,000 because I wouldn't be paying my mortgage or the electric and the utilities over here at my house where I own. And I would just like again, I would probably rent this place out and just keep it in the you know, as a property that I own.
Yeah, that makes sense. Yeah, there's definitely a lot of nice facilities out there that are for sale right now because of how much they've squeezed the licensing. They really have
Yeah.
done all they can to suppress the licensure quite a bit there.
So I'm thinking maybe March or April, I will be looking. I'm gonna get the machine that I need to manufacture those, regardless of like the time frame in which I'm able to do the farm project and get that going. Um, but I'm gonna probably be start looking in April at getting all of the equipment imported, so that way I can start manufacturing those at scale, those NutriGrow pots. And then not only that, there'll be multiple sizes. So doing things like seed start trays, where you can just put it into like an inert media like cocoa, and then the pot will actually deliver the nutrients.
Yeah, I know you spent a lot of time and research developing these. They're really cool, and
I did.
they're I'm really excited to see how they work out.
Yeah, I think that's a game changer for agriculture altogether and horticulture if you're trying to eliminate single-use plastics and if you're just trying to create better logistical efficiencies because these things contain enough fertilizer that if you were growing like specialty crops or corn or soy, you could start your seeds with these and plant with your seed and you wouldn't have to use starter fertilizer at all. You probably wouldn't you would here's a good example. If you had a 20-gram pot and you put in 50,000 of them in a hectare of land, you'd be adding 1,000 kilos of fertilizer, which is probably enough to be sufficient for that whole entire crop.
Nice. That's really cool.
Yeah.
Sweet. And I know you've been also working on a bunch of different strains for people to put in those pots. You want to tell us a little bit about some of the different cultivars you've been working on?
Yeah, I've been
Excited for for this year?
doing the blueberry train mac. That's, a project from the, last 5 years. I just continue. I'm on F4s now. But I'm still also hunting through some of the F3s and just, double checking stuff. I have to go through these F4s to see if the male that I use from that F3 population is passing the right characteristics and I'm getting the right profiles and everything's kind of homogeneous. So I've got that. I crossed the Chem Dog into Capulator's gas, which is Starfighter crossed to Mac. And I was expecting, fuel, sours, chem, and I am really, really surprised because what I'm getting is this really, really bright, sweet, sweet kind of jackfruit profile, and it totally threw me off guard because I wasn't expecting it at all. It's totally different than both of the parents. So I thought that was kind of cool. I'm making F2s with that because I didn't release the F1 population because I didn't have enough pollen. Uh, that was the last bit of pollen I had from that male. And so I wasn't able to make a ton of seeds. So I'm just running through some phenos of that right now. What else do I got? I have the Gay Laser and the Blueberry Starliner in testing. Those seeds will drop in about 4 weeks or so. And the Gay Laser is Sunset Sherbert and Rainbow Belts crossed to that Starfighter Mac. And then the Blueberry Starliner is the Trainwreck Blueberry MK Ultra crossed to that Starfighter Mac. And yeah, dude, I just been popping a bunch of seeds because I bought a couple of those like Vivo Sun 2x2 tents, and I can fit 9 square 1 gallon pots in those. So what I'll usually do is I'll pop about 20 seeds, and then I'll sex test them, take the best nine, load them up, and I'll rotate different, seed stuff every couple of weeks. So it allows me to pop more seeds consistently and rotate them. And they're not like really big hunts, Obviously, I can only pop about 20 seeds at a time for each tent to pick the best nine. But um, it's allowing me to go through stuff a lot faster now that I'm like rotating them like that. And I can always just run more of the same stuff, like because I have multiple tents. So if I was like want to do 60 seeds, it's fine because I can rotate them through the different tents. And then anything that I really like, it gets run back in the big room in my soil beds. So like I'll find cuts and I'll be like, dude, I gotta run this like regular and fill up a bed and see how they do.
Nice. That's really cool. Anything you're particularly excited for this year for doing your, um, anything once you get your facility going? Do you have any specific ones you're excited about or excited to tell people about?
Well, Battle of the Breeders is one of the things that we'll be doing out of the facility. So, one of the 4 light rooms that we have is what's called Battle of the Breeders, and essentially there's 4 lights, right? And under each light is an autopot. We're using autopots in that room. They sponsored that room, and I have 9 um 3 gallon pots or 2.9 gallon pots under each light. So what we're doing is we're doing 4 breeders per run and 9 phenos of one variety per breeder each light. So over the course of the year we'll have uh 16 different competitors. And what we'll do is, I'll pop, however many seeds, probably I think like 20 seeds of each variety from each breeder. And then we'll pick all the best nine, like I'm kind of doing here. It's going to be a much larger scale, bigger pots, bigger plants, bigger lights, bigger room. And then we're putting the phenos together in a basically a bundle, like where we have a box for each breeder. So like we have Capulator sent something. He sent like a apricot or an orange something. I can't remember exactly what he sent, but he sent me something. And he's like, "Here, run this for the Battle of the Breeders." So at the end, when we harvest and we have nine cultivars of 9 phenos of every variety, we'll have a 9 gram like quarter box. It'll have some cool stuff in there, probably like a grinder, a tray, some papers. It'll be all co-branded. This is the Capulator box, and there'll be 4 boxes from each different breeder that we do drops on. Everything else in the facility is always going to stay the same. Same standard 6 flavors. We have our uplifting varieties. We have our mid-range varieties. We have our sedative varieties. So we'll have 6 in-house things that always stay the same. But the Battle of the Breeders will be specifically just for collabing with different breeders. We have Fresh Coast on there. We've got Third Coast, which is max yields. We've got TH Seeds. We've got 808 Genetics. We got Northern Lights Research Group, which is Greg McAllister. We've got, who else is in there? Capulator's in there. Mean Gene is in there. Like, we have some of the best. And the thing is, it's also not only is it just a battle of the breeders where we put the boxes together and we do the drops, but it's filmed. It's going to be a podcast series called Battle of the Breeders. So we're going to be doing breeder interviews. We'll be featuring each breeder's variety on the show. We'll do educational tips and tricks. We'll do little features and stuff. And so what we're trying to create is an ecosystem around our dispensary and grow where it's not just a vertically integrated business, but we're also connecting with the community by showcasing other people's work in the community in a really like high efficiency, well managed, data driven cultivation setting that somebody who may not have flower in the state of Oklahoma, like, Fresh Coast or something, they can say, "Hey, look at dude, this is we bred this. They're gonna do a drop of something that they hunted out and grew themselves. And if you guys are interested, creates the spirit of like collaboration and promotion. Like we can cross promote. I can help promote other people, and I don't know. It's just it's a really I thought it out very extensively, and I just wanted to create again like an ecosystem that isn't just another cannabis dispensary, right? It's not just another grow. It's something that the community can get involved in, and something that like can drive. It can help drive sales, but it can also help like showcase like what the culture is when like people that are from the culture are doing things.
Sure. And you had mentioned like about having the different profiles you're looking for your dispensary. You mentioned like sedative and a couple of others. What are the different like profile types you're aiming for your menu for the dispo? Like I know sedative ones and ones that help people kind of sleep or deal with pain is definitely one of the biggest ones. But like what are the ones that you decided on, and then how did you kind of come to that decision? Because it's always interesting to hear people's perspectives on that.
Well, we haven't fully decided on the menu, but I definitely know Chem Dog is going to be on there. That'll probably be right in the mid range because that one for me at least is not super sedative. It's not super uplifting. It's just like perfect right in the middle. I'm definitely gonna put like a haze in there, maybe like Jack. Maybe put like Jack. Or put like a super silver. I'm definitely gonna have a pheno of blueberry train mac in there. We'll probably have like a Kush, like a PK or something like that if we can get a hold of, if I can find a good cut. Probably Blue Dream, even though I don't like Blue Dream. But I know how popular it is, so probably something like Blue Dream. and then probably something like up that's more modern, or like middle school, like Gorilla Glue, right?
It makes a lot of sense.
Yeah.
Um,
I'm thinking like between Gorilla Glue and Mac, probably Gorilla Glue over Mac, but then we got to have a couple of a couple haze varieties, got to have some Kush in there. Just have a good a good blend. I don't need
right.
31 flavors. I don't want to try to be Baskin Robbins, I just want couple of things that are done really well, that are consistent, because here's the thing, when I was back in the trapping days, it's like I paid my bills with like certain varieties for over a decade, like the Purple Kush that we had. I mean, we ran that for like 15 years, and it's like nobody asked us to switch it up. That was the one it always sold. And it was because we always it was the same thing. People liked it. It was consistent. And that's the thing. When people find things that they really like, they always come back. I mean, people don't buy McDonald's because it's good quality. They buy it because it's consistent. And I'm not saying that we don't have good quality because obviously the goal is to always be producing the best possible that I am capable of. But consistency is so important, for building a brand.
Absolutely, yeah, for sure. And then, like you were saying too, the purples always tend to fly off the shelf more than everything else. I know there was a used to be one way down in Hugo or in the part of the southeastern corner of Oklahoma, and they had like 200, 250 different strains, and they would always sell out of their purples, just like you were saying back in the black market days. So like, doesn't matter if you have a shit ton if they're just gonna sit there, just like you're saying, you're better off focusing on
The
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 hard
purple
hitters.
cut that we had, I think, is what is called the ultraviolet. But it was very trichome laden, and the purple were like hues that were streaked through it. So it wasn't like that anthocyanin purple that was really dominant in the inflorescence, but man, it was like an OG, but it wasn't like, it was weird because it grew like the Master and the Bubba. Like you couldn't tell them apart until they were like maybe 5, 6 weeks in flowering. But the PK, it had a different nose. It wasn't that earthy coffee. It was more of like a OG type nose. It had a little bit of like sweetness in it as well, a little bit of like almost kind of grape notes.
Awesome. No, that's really good. I look forward to kind of seeing how that works out. That's really
Yeah,
great.
it'll be awesome, man. I can't wait.
Sweet. What are any takeaways from last year? Just talk. I mean, you talked to a lot of different growers and everything. Any different trends that you think people need to be aware of, or
Yeah.
especially pest control stuff too, as far as like new issues that you saw or issues you saw more often?
Yeah, you know what? Last year I went to CanMed out in Puerto Rico, and I went and I met up with Kevin McKernan, and I was talking to him about this leaf spot pathogen, which I thought was originally, when I had identified it, I thought it was leaf spot septoria. But then I started noticing that this same leaf spot was affecting things like grass and other species. So I was like, no, it can't be, because cannabis leaf spot septoria is specific to cannabis and it doesn't hop from different varieties. So I was like, we're dealing with something else. And so I was talking to Kevin McKernan about it, and he's like, let's do a genetic essay and, send me some samples. And then, so I sent him samples of our spores, the pure spores that we have, the trichoderma, the bacillus, and I sent him samples, and they did a thing, and they identified it as alternaria. And then we did a follow up with Cove Environmental and one of the professors of biology at OSU to do an ionomics project to just see what the ionomic makeup, or the total ion biome, which is basically all of the elements that make up, and how that affects how that pathogen is able to bypass defense systems and become more virulent. And we were looking at different elements that could possibly be present that could cause that. And so things like, I'd have to pull up the paper, but I think there was some heavy metals and stuff that were when in higher levels. Things like, I think, like some micronutrients, when they're at higher levels, it shows that the pathogen is more prevalent in those samples, in those leaf tissue samples. And so, I want to continue doing research on this, because what's interesting is, what we saw is the majority of the Alternaria is just a strain that's ubiquitous in the environment that's kind of just opportunistic. It's not really virulent, it's not pathogenic. It's just, but there were some other species in there that were virulent, that were pathogenic to plants. And so what I'm thinking is the ionomic makeup, or what the plant has in its total, makeup, and how that's affecting these microbes, how something that might just be opportunistic might change to something pathogenic because of the amount of nutrients or heavy metals or X, Y, and Z that might be present in the tissue is changing the actual transcription genome of that microorganism to be able to bypass plant defense systems. And and so I'm questions that I have are like, okay, we're seeing, multiple different species. Is there some type of, mechanic at play that's involved with nutrients and different elements, or is there horizontal gene transfer from one to the other where they're picking up these things, and then this larger group, which isn't necessarily pathogenic, is becoming pathogenic? And how are these things functioning? Because having answers to those can help us, mitigate those problems in the future so we can avoid these issues. And, one of the problems with Alternaria is that it kills the plant. Like, it doesn't care about keeping its host alive. And the candida, or the spores that form, they're able to survive composting, even thermophilic composting. They can live on dead, decaying organic matter. They can lay dormant in soils, and the way that they spread is really easily through water, through the air, um, through physical contact, mechanical contact. And so it's much more harder to combat this particular pathogen than something like leaf spot, septoria, or even some of these other common things like powdery mildew, right? And so what we have seen in the last couple years, especially out here in the Midwest, up in, places like Michigan, Chicago, Ohio, Oklahoma, Mississippi, is we're seeing this Alternaria, this leaf spot pathogen, become more widely dispersed, and it's causing more damage to more people's crops nationwide. And I believe it has a lot to do with the fact that, because it can survive in the compost, and a lot of us, especially organic farmers, are composting biomass and doing all these other things and using this compost on our farms. And also because it affects other plant species, just treating the area or your plants directly for this pathogen isn't enough. You need to treat the grass, you need to treat the trees around your property. You have to like take more measures to combat this problem to prevent it from taking your crop out. Because, dude, especially if you're in climates where you get, rainfall, dude, after rainfall, dude, these pathogens just explode. And so having preventative measures, using things like the microbe spores that I have on my website, the Bacillus subtilis trichoderma combination, even Beauveria bassiana, they were showing um, can do some of these things where it prevents pathogens, and it works as an endophyte to protect the plant internally. And some of that data, which I've been talking about for years and years, was confirmed recently at the CanMed conference just last year. So I felt that was really validating. But yeah, we just have to take more preventative measures for these things because they're so widely dispersed and they're getting worse and worse.
I know when I was in Oklahoma, first off, shout out to Kevin McKernan, super big friend of the community. I know I've sent a bunch of samples to him over the years, and he's been on the show a bunch of times. If you want to check it out, he's got a lot of great white papers he's worked on as well on plant pathogens, especially on aspergillus. He really busted that one wide open and showed how there was even some biocontrols that were causing false positives with aspergillus. It was at a
Yeah.
Empicoliensis was causing it. Bacillus empicoliensis was causing false positives. But I noticed when I was working in Oklahoma, the places that had really good mature soil, like more compost, or places that had not had chemical ag, tended to not have as much issues with it. And then the places that were like getting hit hard, the hardest, were places that were converted cow fields or cornfields or wheat fields where they sprayed 2,4-D or some other, heavy herbicides that just annihilated all the soil fungus so that there was nothing else to like eat those spores in the winter and kind of have some competition going on that. And
Yeah.
not to say that that's the only cause or that you'll definitely have that, but it tended to be like 80-90% of the fields that I did see it were ones that were a year or two beforehand had been treated with these horrible chemicals that you should never use around cannabis. The other thing, like you were saying too, is that the both Alternaria and Septoria both tend to spore more after the rains and 85 plus 80 degrees temperature. So if you have that combo, that's really when they kind of go to spore the heaviest. Not that they won't otherwise, but it tends to be where they're doing the most. And then I've also seen, just on to like further support what you were saying, Jadam preparations, especially Jadam microbial solutions and Jadam nutrient solutions. I've seen spread Septoria and Alternaria, pretty quickly through a grow because they
Yeah.
had a contaminated leaf
Yep.
mold or whatever, and then
Absolutely.
now it's on everything because you just inoculated everything.
Yeah, and a lot of people don't realize this, but if you're ever having a problem and you don't know, if you have a balanced microbiome and you feed complex carbohydrates or you try to make, ferments or solutions that are contaminated, you're going to procreate those bad microbes and then distribute them even further. And so it's really important. It's one of the reasons why I tell people that you shouldn't be feeding your soil molasses, because they're like, oh, well, feeding well, a lot of people think that feeding their plant. And it's like, no, the plant doesn't feed on carbohydrates. It manufactures carbohydrates. And then about 90% of those, 85 to 90% of those are dumped into the soil through root exudation to stimulate the microbes. But when you just dump in a heavy concentration of carbohydrates, it's going to rapidly increase the microbial proliferation. And if you don't have a balanced microbiome, then things can get out of balance really quickly.
Oh yeah, and that's why I always tell people to stick to either inoculations like the ones that you have available, or Korean natural farming where you can visually see that it's a white, healthy colony, not
Mhm.
just a random leaf mold that might be fuck knows what. I
Yeah.
think it's one of the things that I think people really don't understand when it comes to some of the natural farming, or even combining natural farming with inoculated, purchased inoculants. You really should stick more to the KNF side than the Jadam side, at least for the microbial stuff. Like some of their pesticide stuff is great, actually. I highly recommend their pesticide book to people, but it's a different. You're not using microbes, in Jadam with that, primarily, except for labs. So definitely is a big point. What are some of the other things that you observed? I know you had done so much research with that. What are some of the other cool things that you learned last year?
Well, what was really cool is I found a plant. It was actually that Starfighter Mac. I found a pheno that was seemed to be completely resistant to I mean, it just didn't have a lick of Alternaria. It didn't have any, spot pathogens, and that's one of the reasons why I used that for some breeding work because I'm looking at being able to breed plants that have a natural immunity or that have higher defense systems built in to combat that issue. But I mean, other than that, we're going to be working this year on the pesticide registration. So we've been doing some research and I was under the impression that it was going to be about 150 to $200,000 to register microbes as a pesticide, but we found some stuff doing some research recently that they qualify under like a different category because they're less harmful and stuff like that. And so like the licensing fees and the efficacy studies, they're not as extensive as like a chemical pesticide. And so it seems like it would be reasonable for us to do that. So we're going to be looking at registering the Beauveria bassiana, the Bacillus thuringiensis, and the Metahar xanthium. This we're in the process now. So we have hopefully that doesn't take too long. I mean, it could take up to a year, but hopefully it doesn't. But we'll be working on that. Same thing as always doing, agronomy work, looking at data, adjusting soils, that's kind of my thing and you know.
Any interesting observations in terms of the minerals or dosing or anything weird that you found last year?
No, um for my outdoor, I use the Humates and Microbes Calcil program. I have pretty poor soil where I'm at, and I've, slowly built it up over the last couple of years. It's still not, you know, where I'd like it to be, but I still grew, 14, 15, 16 footers. I'm not going to be doing that anymore. I'm going to start putting plants out later. They're just too hard for me to manage them. I didn't top any of them. I didn't train any of those specifically because I wanted to look at the natural structures because I was doing a giant hunt. Um, but I'm not gonna. Other than the stuff that I mentioned about getting a farm, doing produce, I still do all that same stuff at my house until I move. So growing potatoes and onions and
What are some
um,
of your top veggie varieties that you like? I have a huge collection of veggies and herbs, but it's triple what
yeah.
my weed collection is. What are your favorites?
Yeah, so I like to grow things that I eat a lot of. So potatoes, onions, garlic. I'm gonna do a bunch of tomatoes and peppers this year, because I'm gonna try to see how that business model would look like on a small scale, just to see if I can get some small customers, farm to table stuff. I have plenty of room where I could grow hundreds of pounds of tomatoes and peppers. And I bet you I could make monsters too, 15, 16 footers. Easy. I can do it with herb. I can definitely do it with tomatoes and peppers. So um but my goal really every year is to grow all of the vegetables that I need to make Havos Rancheros from scratch. So I grow peppers, onions, tomatoes, beans. Corn for me is difficult where I'm at. I don't have I've got really poor sandy soil, and it's really hard to get like adequate zinc levels and like the micronutrient levels like on point that corn needs. But I do try. But yeah, that's what I grow. I grow, and then loofah. I grow loofah every year, and some stuff like that, cucumbers.
Any new things that you've been researching that you're excited about, or um, interesting rabbit holes you've been down lately?
No, not so much, man. Like I've the when it comes to the agronomy stuff, man, I feel like I really got all that nailed. Every once in a while, I'll run into some stuff. Like I was reading a paper on iodine the other day, and they were showing that you know iodine actually has a is could potentially be considered a beneficial nutrient, and it helps increase biomass, and it showed some other things. I just briefly skimmed through this paper that I printed out the other day, but I mean nothing that's like excited me.
Nice. I've been tinkering around with lactococcus species lately. You can do bladderwort or butterwort inoculations for lactococcus, which complements lactobacillus. So it's like a way, something else to add if you're like trying to really smash down fungal infections and stuff like that.
Mhm.
That's been kind of fun to play around with lately. So, what are some of the other any other trends that you noticed last year with people doing stuff wrong? I know you get a lot of questions and people asking for help with the shop and all.
Man, you know what? We built a great community around, our social media, YouTube and Discord. And honestly, I see so many different people's grows, and I mean, I shouldn't be, but dude, I'm like, sometimes I'm shocked just to see how well people and how quickly people progress when they have access to the right types of information, the right community, and you have like people helping you out, answering questions. And I see so many people do so well with so much less than what is conventionally used in the space. So other than just like getting more people to grow and getting people better at growing and sharing my knowledge, sharing my genetics, that I mean that's what I see. As far as like new trends, I don't really pay attention to like hype stuff or like, because I'm like pretty set in my ways, and I've gone down all the rabbit holes, and every once in a while I'll come across something that's like oh, that might be cool to implement, and I might try it out. And if I don't see a net positive benefit, it just kind of goes to the wayside. Like chitin, right? I like there was a company that had sent me some liquid chitin extract, right? Like, and I used it in my system and stuff, and I didn't see any. I couldn't tell any difference from anything, right? So it's like I could add this. It's not hurting anything, but I'm not noticing any difference. So it's like, all the other biostimulants are probably already enough. The humic and fulvic acid, the amino acids, the Kiaha, the microbe plus, the aloe vera. I think just another thing on top of it isn't really making any type of difference. So it's like this stuff like that I'll play around with. And same thing with like silica. So, a company from the Netherlands, they sent me a couple bottles of some soluble silica, and I already run things like the calcium silica and the microbes and stuff, and I have really good, so I used it. I couldn't see any definitive benefit because I think it's probably because I already have that in my system through other means, which are one more cost efficient and more efficient from a resource standpoint and everything else. So it's like I tried little things here and there, but I think the program is like my whole program is basically a biostimulant sandwich that interconnects plant nutrient with carbon dynamics. It interfaces. It gets that basically the nutrients get that software upgrade with all the carbon and microbes and everything. So there's just really I don't feel like there's much more that I could do. My soil structure is good, aeration, the water, the nutrient density, the balance of nutrients, everything is just good.
Nice. Do you want to explain a little bit on the amino acids and the fulvic and humic? Yeah. You
Yeah.
and Dr. Fausto have been some of the best people I've heard to explain those.
Yeah, well, let's start with amino acids. I actually just did a podcast recently on amino acids. I've been yelling from the tops of my lungs about amino acids for years now. Amino acids are really interesting because typically when you look at conventional fertilization practices, you're seeing nitrates or ammonium applied as the main sources of fertilizer. In organic agriculture, typically you're seeing organic meals like soybean meal, blood meal, feather meal, etc. Um, which also break down into amino acids eventually through microbial metabolism. But when you deliver soluble amino acids, what it does is it acts as a biostimulant. So all of the downstream effects that you get from the proliferation and increase in microbial diversity and the metabolites, the enzymes, the nutrient cycling capacity, the biofilms, the organic acids that chelate things, those all get kicked into overdrive from a microbial side. But then from the amino acids themselves, things like glutamate and glycine, they chelate cationic nutrient elements. So they can bind with things like calcium really, really well. And amino acids have shown in the science to be able to increase calcium uptake by up to 1,000%. It's insane. But they also bind with other metals like iron and zinc and manganese, and they chelate them, keeping them from oxidizing or keeping them from different precipitation reactions. And so they stay in a metabolically available form that can be used for the plant. And when they're chelated to organic carbon, it can bypass any type of um, active transport systems that the plant might need to use energy to uptake those things. And then one of the things that's really cool about amino acids is the energy dynamics because amino the plant has this natural cycle and it can take up to 18% of the plant's metabolic energy to complete this cycle. But what happens is the plant basically, for the most part, takes up nitrogen and nitrate as a nitrate, and then it's converted internally into ammonium, and then this ammonium is converted into amino acids, and then those amino acids are converted into proteins. This is a highly energy intensive uh, process And if the plants are able to take up the amino acids directly, they can skip some of that conversion process, the process where the nitrate is converted into ammonium and then ammonium is converted into amino acids. So that energy intensive process is bypassed, and that energy can be used for other metabolic processes such as defense systems or whatever else, terpene expression, whatever else it is. And so amino acid nitrogen is massive. And then there's also a paper, science white paper, it's called Catabolism of Amino Acids. And what it also shows is that plants can catabolize stored amino acids for energy in the form of NADPH, FAD, and ATP. And so all of these things are responsible for biological energy exchange and for all plant metabolic processes. And so if you're in a situation where the plant's under stress or it doesn't have access to phosphorus to make ATP, or it's in some type of energy limited system, the plant can actually start breaking down these amino acids to keep these internal processes functioning empowered.
Well, that's really cool. And I know I've definitely seen a big difference adding humic and fulvic acids to aquaponic systems too. And
Yeah.
they not only helped with the plant growth, but they helped with the fish growth as well, which is kind of surprising. When we did a side by side, it was even the fish were coming out 5%, on average weight heavier. So it was kind of interesting to see that it was helping the gut biome of the fish as well, which
Yeah.
is kind of
Yeah.
interesting in a quantifiable way.
Well, humic and fulvic acids, to clarify, there are actually different classes of molecules that are defined by their function and their weight. So humic acids are a larger molecule, and they can help with things like soil structure and again, downstream effects. Anytime you improve soil structure, you might be stimulating gas exchange and water dynamics. You could be influencing the microbiome. It helps with long-term nutrient holding capacity by increasing uh, cation exchange capacity. Humic acids is good for tying up heavy metals because it'll bind with heavy metals and keep them insoluble. Um, Humic acids isn't great for trying to deliver nutrients because it holds on to them and it doesn't deliver them in a soluble form. But fulvic acid is a lower weight class of organic molecules that chelate nutrient ions and keep them soluble across all pH ranges and allows for them to be delivered more efficiently through the cellular membranes. And when you think about fertilization, fertilization happens on a cellular level. So when we're feeding ourselves, our stomach, for instance, what those acids are doing, what those microbes are doing, is they're breaking down the base chemical constituents and they're feeding the cells that way, right, in our bodies through our bloodstream. But when you have humic and fulvic acids, or especially fulvic acids, you can deliver that nutrition through the cells more efficiently. So whether it's fish or humans, or if it's in the soil, that fulvic acid is binding with those nutrient ions, and it's creating bioavailability with those things. Just like the amino acids, they chelate nutrients and keep them in a metabolically available form for the plant. And then the fulvic acid is small enough that it can actually be used as a carbon nutrient source for the proliferation of microorganisms as well. So they act as a biostimulant, but they also act as nutrient chelators and basically they interface with nutrients as a organic delivery system. Because when we're talking about things like mass flow, um, root inception, and diffusion, there are different forms. There's active and passive transport mechanisms. And when these things are attached to the organic carbon molecules, they can be passively transported, so the plant doesn't have to expend any energy to uptake those. And another thing, too, is when you think about the nutrients that are being delivered, the majority of those are cationic, right? So they have a positive charge. And it's like a battery. You can't just always charge a battery. At some point, you either need to use some of that juice, or there has to be negative charge put in there if you're going to keep putting more positive charge. And so when you're delivering those cationic nutrients, it neutralizes some of that charge so that the plant can actually balance its sap more efficiently and can take up more nutrients because it's not affecting that sap balance, right? So what'll typically happen is if you're not if those aren't carbon chelated nutrients and it's just positively ionic nutrients, the plant will use its potassium sodium ion pump to release either sodium or potassium so it can take up other positively charged nutrients that it needs. Or things like phosphate, sulfates, and nitrates will um, will kind of level out those positive charge because they're neutral, so the plant can keep taking those things up. So there's a massive interface when it comes to carbon dynamics and how these nutrients are bioavailable, how they're transported, and how they actually function from an energy standpoint within the plant.
That's really cool. I know some of the organic people too on the aquaponic side of things and organic hydro do use the fulvic chelated iron, which is something that a lot of people haven't a chance to check out either. But
Here,
I
look
know
at
that,
this. Check this out.
yeah, there you go.
I just
That's
recently made some myself, and I did a little quick YouTube video. But what I did was I took citric acid, I took absorbic acid, which is vitamin C, and then I took pure fulvic acid, and I took iron sulfate. And what I did is I mixed up a solution with the citric acid, and then I used the absorbic acid because that's an antioxidant, and I got the pH solution down to 3.5. I added in the pure fulvic. I divided them up into two separate containers at 3.5. I added the fulvic acid to one and then adjusted the pH to keep it at 3.5. And then in the other one, I added a solution of iron sulfate, citric acid, and absorbic acid. And then as I added the iron sulfate, I adjusted the pH down to 3.5. Then I combined both together and I agitated them for a little over an hour in a blender at a low pH range. So that way the pure fulvic acid would bind with the iron under reduced conditions, and the only thing I need to do is a jar test now. So my next thing for my next experiment is just to take some phosphate, maybe from like monoammonium phosphate or something like that, put it in a jar with the iron fulvate and keep it in that jar and watch for precipitation reactions. So if anything precipitates and there's any type of residues that fall out of solution, then I know that I didn't get a good enough chelation, or maybe I used too much iron sulfate. But I've already made this and I've already fed my plants with it, and my plants responded really, really well. If you're ever having any type of interveinal chlorosis, which looks similar to magnesium deficiencies, but it's at the top of the plant as opposed to on the older growth, it's typically a sign of iron deficiency. And iron deficiency is really, really easy to come by in organic systems because you need a really low pH or an anoxic soil for iron and things like manganese to be available. They need to be in their reduced form, and we're usually not working in those conditions, and so it's usually one of the most limiting nutrient in organic agriculture. And that's why I recommend things like the Bacillus subtilis and trichoderma, which create the hydroxamate sidiophores to grab onto iron and chelate it. That's why I recommend the fulvic acids, and it's one of the reasons why I made this up, just to try as an experiment to see if an exponential increase in, well, not exponential, but like if we have better solubility of a carbon chelated soluble organic iron source, as opposed to me just doing my standard protocol with the microbe program to get iron more available, if we're going to see an increase in biomass and fluorescence, or if we're going to see any type of effect due to having a higher quantity of that nutrient available.
Yeah, and that's a great one to use in aquaponics, especially, and iron's one of the first things that goes deficient in aquaponics. It's usually iron, then manganese, then molybdenum, in that order, in aquaponic systems. If you're maintaining your pH at least.
Yeah. Yeah. Yeah, and that's the most difficult thing about micronutrients is that all of the micronutrients, with the exception of molybdenum, um, they need to be in the reduced forms in acidic soils to become available. And typically, we're not running our pHs that low. And so we're like, how can we increase these? That's one of the huge benefits of using things like the Micro Plus, the probiotics, the microbial inoculants, is because, like, let's say Micro Plus, when you water with that, it's got a lower pH. You don't, and you want that lower pH because that solution pH is going to have a higher availability of those micronutrients, and your media is going to buffer it. So it's not like it's going to change your media pH, but it is going to change the availability for that short period of time when you're watering of those micronutrients, and the availability for the plant and the ability for the plant to access those.
Absolutely. And one other thing, just because we're talking about molybdenum, if you are running hydroponics or aquaponics and you're using nitrate as your main nutrient source, you need to actually boost your molybdenum quite a bit above what you normally would for a soil system, because the plants are going to plow through that molybdenum much faster because they need the molybdenum to convert that nitrate back to a usable form of nitrogen. And it's one of the things that's like almost never talked about in aquaponics.
Yeah.
And it's like critically important to like maintaining your system, because like 100% of the clients we've picked up doing the aquarium maintenance, aquaponic maintenance stuff that I work with True Aquaponics. Every single farm we've ever tested that was a new client was almost at zero molybdenum. And a lot of them are like, oh, well, my red lettuce isn't red anymore, or my purple cannabis isn't purple anymore, or whatever else. And it's like, yeah, well, you need the molybdenum also for your anthocyanin production. And you can even force an overproduction of anthocyanin with a mild, emphasis on mild molybdenum.
Yeah, it's super easy to overdose on molybdenum because the levels that are needed for plants is so low.
Yeah.
But like you're saying, the main job of molybdenum is for nitrogen conversion. That's its main job. It's to literally that process that I talked about where you're converting ammonia, nitrate into ammonium into amino acids. That's molybdenum doing its job.
Yeah, absolutely. Yeah, no, and just like you were saying, that's why it's so important to get, inoculate your system with the right microbes and make sure you're maintaining good balance, because if you don't, you'll absolutely not have these different processes that can convert those things like iron and molybdenum back to something that plants can actually use. I think
Yeah.
that's something that people don't quite understand, is like, especially a great example of this too is if you like Mulder's chart is another one where you might have a severe iron deficiency. Well, if you have a severe iron deficiency, it's going to cause phosphorus deficiency. And you're going to look at your little color chart online and go, oh, well, my leaf looks like it needs phosphorus. So you're
Hmm.
going to go add phosphorus, but now your phosphorus to iron ratios are even more off than it was before, and it doesn't
Yeah.
actually fix it. So it's one of the things
Yeah.
where if you have your right microbes, you don't run into some of these issues, and then
Yeah.
have to go and use Mulder's chart or some of these other tools to try and figure out what the heck is wrong.
Most people look at a Mulder chart and have no idea what they're even looking at. I know exactly what I'm looking at when I see a Mulder chart because I understand how all nutrients interact with other nutrients back and forth versus which ones are, you have like the calcium boron relationship, you have the nitrogen molybdenum relationship, you have the calcium nitrogen relationship, you have the relationship between phosphorus and every other single cationic nutrient, depending on pH. You have pH dependent reactions, you have non pH dependent reactions. There's like all these different things, and um, it can be really daunting to try to understand the Mulder's chart if you really don't have an agronomic background.
I always explain it to people as like, think of these as different little points or weights on a plate that you're spinning on a chopstick, right? You have to keep them balanced or it'll wobble and fall off the chopstick. And it's like a super simplified way. I mean, it's super
Yeah.
ultra simplifying it, but it basically says, okay, well, if they're a stimulant and do stimulation, then you need to add those in ratio. And if they're an antagonist, then you also need to consider that you're affecting those other ones in a
Yeah, and
negative
there's a
way.
lot of things on here that you don't see, right? Because you could draw an arrow going towards manganese from iron and an arrow going from manganese towards iron where they converge and point at each other, where it's going, oh, these two things don't work. These things are antagonistic to each other. Or you could have arrows from calcium, potassium, magnesium that show, hey, these are so there's even
Yeah.
more than just this.
Oh yeah. And there's ones that have selenium and cobalt and
Yeah.
iodine, and you can get way deeper down the rabbit hole than this on Mulder's
Yeah.
chart. There's
Yeah,
many,
and
many
that's one of the
versions.
things that people don't like. There's a couple of nutrients. So nickel, cobalt, selenium, iodine. are four elements that aren't really talked about very often in agronomy. But if you look at some of the science behind those nutrients, they've shown in, model crops, Arabidopsis thaliana, which is usually, what they use to do a general study with, that a lot of these other nutrients that aren't often listed do have can have significant impact on overall health and quality of plants.
Oh, absolutely. Here you go. Here's one that's a little more fleshed out.
Oh yeah, yeah. Yeah, see, they get, and these interactions can get really confusing if you're looking at this going, what does this even mean? I mean, most people don't even know that, if you're looking at it from 12 to, going clockwise, that we're looking at phosphorus, chromium, copper, lead, iron, selenium, nickel, calcium, gold, cadmium, Hg. Ooh, that's one. Hg. No.
Mercury.
Yeah, mercury. Aluminum, copper, manganese. I know what all these elements are. I can look at most of the periodic chart of elements and tell you what the element is by its abbreviation as well. But I'm a nerd, you know what I mean? I'm a
It's
science
just
nerd.
it's just one of those things that I think that people don't realize. Like, if you are having a weird deficiency issue and you add something and it doesn't fix it,
Yeah,
this
yeah.
this is the thing to turn to try and decode what's going on.
Yeah, I just did a podcast on magnesium, and we're talking about how magnesium is like one of the power energy gatekeepers is how I say it. And it's because even with proper ratios of NPK, without proper ratios of magnesium, um it's like running the factory with the lights on but without full power. You know, it's needed for phloem loading. It's the center of chlorophyll molecule, so it's responsible for photosynthetic efficiencies. It's, it's used to activate more enzymes and coenzymes than any other nutrient in plants, over 300 different enzymes.
That's awesome. Yeah, no, I didn't know it was that used for that many different enzymes. That's awesome.
Yeah.
Well, cool. Well, I don't want to take up your whole night. Is there anything else you want to mention here before we start to wrap up the show?
Yeah, so code save now, one word. It's a one-time use code for 20% off on the website. And it's not just new customers. You can use it one time. Anybody can use it. And also, Greg McAllister, aka Seattle Greg, the man who popularized Northern Lights by bringing it out to Amsterdam, who currently resides and have been doing breeding work in Hawaii for decades now. Um, he sent me a bunch of sawtooth freak show hybrids to just give out to the community. So those are also going out with orders until supplies last. And he gave me quite a bit. So I've just been throwing those in packs. So go tap in there, support small businesses, and you know, sign up for the newsletter, man. We have so many good resources, and we're here to help you guys grow.
Those freak shows do really good in full sun. You can put them in the full Oklahoma sun with no shade, and they'll rock it. Same thing. They're the only ones that really tolerated the full sun, except for some of the sativas. But of the like more work genetics, they were the only one that you could put out in Thailand, and it would just take it. You know what I mean?
Yeah, nice.
So they're great for that. The other cool thing we found in the freak show line in Thailand, we found one or two plants that re-vegged on their own, kind of like that like mythical Reunion Island plants. I have
Wow.
some Reunion Island seeds still I need to back
You have a
cross.
perennial? That would be so awesome if you could
But
breed a perennial crop, bro.
so I know someone there that had a freak show, and they had gone through four harvests and hadn't, the plant was just re-vegging itself. So
Wow. That's pretty cool. That'd be so neat to be able to like have your favorite variety and have your spot picked out in your yard and you're like, okay, every single year at the end of the year I'm gonna prune this down, just like you would with peppers or whatever, and then in spring it starts growing again. Dude, that would be so legit.
oh yeah, but I think that the gene mutation that is in the Reunion Island is also in the. I think it's a double recessive in the freak show line, like
Interesting.
based on that observation.
That's something to look into. Did you see the I saw this thing on looking at it was like a quadruple stalked trichome head in I think it was a tomato. or something like that, and they were
Whoa.
talking about how if somebody could isolate that genetic characteristic, they could make like hashers that were exponentially higher in their return rates. And it got me thinking. I was like, dude, at Cove Environmental, they have like they got grant money to build out a state of the art brand new soil testing facility. They can do leaf tissue, sap, soil, they can do genetic research, they can do everything. And so I sent it off to my homie Landon, who's working on his PhD thesis right now. um, and I was like, dude, we could do this. We could figure this out. Like, we should totally pursue this, because if we can figure this out and then come up with a marketable variety that has this characteristic for the trichome head, dude, that would be like insane, bro.
I've seen that a couple times, just scoping plants, and it'll be like one trichome has maybe two heads or something on it, or three, but not something that was stabilized. The only person I've ever seen that I know is playing around with that, and I've seen it like on some of their, but not again, it wasn't the whole plant, but they certainly had it stabilized a little more than the most. Was Bob Hemphill had a strain, I want to say 2019 or 2022 at the Humboldt Regen Conference, had a strain that had that on a lot of the trichomes. It was like, again, it was still not the dominant one, but it was far more than I've seen anything else. He'd be a good guy to talk to on that.
Definitely. I actually reached out to him not too long ago. It's hard to get a hold of him on social media though.
It is. He's a ghost in the hills.
Yeah.
Alrighty, well, thank you so much for joining us. You want to mention how people can find you, and I'll
Yep.
put your links up here on the screen.
Fullcropscience.com with one L, FULCROP sciences. You can also find me on Instagram at rust.brandon. And if you guys are looking for any of those resources, just go sign up on the newsletter. It's at the footer of the website. And we'll send you an onboarding email and get you all set up to, yep. It's all education everywhere. We got blogs, we got YouTubes, everything.
Sign up right here on the website, right at the bottom.
Yep.
Awesome. Well, thank you so much for your time, and I really appreciate you coming on. And then also for those of you watching this, this is going to go live right after the next day after I upload the Wayne Justman memorial video we put together. So I took all the episodes like we did with Frenchie when he passed away. We took all the episodes where we had Wayne Justman on the show and combined them into one. And I got a little intro for it. I think you guys are going to like. So he passed away last week. He was the guy that was medical patient #1 and really did a lot of work with Dennis Perone to get California 215 passed. And he unfortunately passed away of a lung infection last week. So yeah, and if you're in the Bay Area, the memorial service is on Valentine's Day. I don't remember what time, but I'll have information if you're going to go on on my Instagram. So check that out. This will go live before the memorial service. So I'll be sure to check that out as well if you're interested in learning more about him. But he's got a lot of good stories in those episodes about him and Dennis, and some of the goofy stuff that went on with the police and things like that back in the day. So be sure to not miss that one.
Cool, man.
All right, guys.
I'll talk to you later.
Thank you. Take care. Bye bye. And thanks everybody for watching. We'll catch you guys again soon. We will have more regular episodes. We have a bunch of cool new people in the queue with shows. I've been recording a whole bunch this past week, so expect some more regular content from the channel. Thanks a lot, everyone, and have a great evening. And be sure to check out this podcast on all of the different platforms. We're on everything in terms of podcast platforms, as well as Rumble and YouTube and everywhere else. So if there's a platform you think we should support, I know we're trying to diversify, especially given how crazy the world is now. You never know when cannabis might become censored again. So if you know of another good platform as well, we're always looking to get the show on there. Thanks a lot, and have a great day. Be sure to check out our Aquaponic Cannabis Masterclass with over 650 slides of education, as well as videos of different aquaponic cannabis facilities, hands-on video training, and a whole bunch more. You can check that out at apmjclass.com or Aquaponic Cannabis Growers on Teachable. It's been featured in High Times, Dope Magazine, Dude Grows, Growcast, Embracing Organics, Growing with My Fellow Growers, whole bunch of different podcasts and publications, as well as features all different, all the different aspects you need to grow cannabis aquaponics: nutrients, pest management, design, aquaponic cannabis business and licensing trials and tribulations, and all the different stuff that we've gone through, Marty and I, over the years. Microbial inputs and a whole lot more. So be sure to check it out. If you're looking to grow the best aquaponic cannabis, or really just get a top to bottom, start to finish, all things reference guide kind of class, be sure to check out aquaponic cannabis class. Be sure to check out the aquaponic and living soil pest control course at thepestclass.com. We have over 400 slides just on pest management, pest control, all things pest management, be it fungal issues, caterpillars, aphids, powdery mildew, you name it. We have solutions on there, as well as a huge list of beneficial insects with release rates and application rates for how to make your own biocontrols, to a whole lot more, as well as a bunch of cool tips and tricks for identifying pests in your garden early on so that you can catch them early. So be sure to check that out at thepestclass.com. If you want to learn all there is to know about aquaponics, be sure to check out the Aquaponic Masterclass at aquaponicmasterclass.com, where we feature the longest course that I've ever done. I've kind of made a hybrid class from all my other courses, and then added a couple of other cool features and things to it as well. You can check it out over at aquaponicmasterclass.com. We do have over 50 hours of in-depth content on there, including over 1,000 slides in the deck alone. Tons of cool videos, tips, tricks, lots of behind the scene footage from a bunch of different facilities that I've worked on, not just cannabis, but many others. We go over how to grow over 100 different types of crops in depth in aquaponics, with pictures of every single one of those crops growing in aquaponics over the years. Tons of fish husbandry information, microbial information, mineralization optimizations, how to dose your nutrients in your system, detailed dosing guides, starting at 10 gallons and going up to 10,000 in the course as well, for making it easy to do the math on all of your dosing of your different nutrients. How-to guides, tours of different aquaponics facilities, and so much more. You can check that out at aquaponicmasterclass.com. Looking to do more about plant preservation and genetic preservation of not only cannabis, but many other types of crops, be sure to check out Copyleft cultivars. We have a wide range of resources, as well as an AI assistant to help farmers around the world better protect their genetics from corporate exploitation. I am on the board of Copyleft cultivars, and I'm proud to be part of this amazing project. You can check out more information either on Patreon or on copyleftcultivars.com. You can also check out the wonderful natural farming AI that we've put together to help make gardening easier than ever. It speaks hundreds of languages, can help you with all manner of plant problems, from pest control to fertilizer creation to natural farming methods, and a whole lot more. You can check out access to that over on Patreon. There's both free and paid versions of the app, so depending on what level of support you'd like to give us, we do offer early beta versions of our latest releases to our newest supporters, and anyone who backs us. We do offer additional resources as well for people before they're available to others, as a way to help fund the program. Be sure to check out the Growing with Fishes podcast. Over 1,000 hours of educational content. We have content on all things plants and soil science, as well as aquaponics, hydroponics, and much more. You can find the show on your favorite podcast app: YouTube, Spotify, iTunes, Google, SoundCloud, Amazon Music, and a whole bunch of other places. Thank you for watching. Be sure to check out any of the amazing resources that we have available to help people with their garden. You can check me out on YouTube over at Potent Ponics. You can check us out over on your favorite podcast app with Growing with Fishes or Potent Ponics, depending on which app that you're using. Most of them are registered for both, but sometimes the indexing works a little screwy on others. You can email me at potentponics@gmail.com. Be sure to check out our classes as well, aquaponicmasterclass.com, apmjclass.com, thepestclass.com. You can check out my website over at potentponics.com or our Facebook group, Aquaponic Cannabis Growers. Thanks for watching.